A very good afternoon, dear students. I am Professor Dr. Vikas Mishra, Professor in Department of Microbiology at CSJMU Medical College, Kanpur. And today, we'll be discussing about different types of culture media which are available for isolation of microorganisms and how they are being sterilized. So starting the lecture, first, introduction. What is a culture media? Culture media is a nutrient material which is prepared for the growth of microorganisms in the laboratory. It is you're culturing the bacteria not inside the body of human being or animal, but you are producing that bacteria, multiplying that bacteria in the laboratory. Then what do you mean by inoculum? Inoculum is when microbes are introduced into the culture medium to initiate the growth of that particular bacteria. They are called as inoculum. Culture, the microbe that grow and multiply in or on a culture medium is to referred as culture. Then what do you mean by cultivation? Cultivation is a process of growing microorganisms in culture by taking bacteria from the infection site, that is in vivo environment, either human or animal, by some means of specimen collection. It can be respiratory specimen in the form of sputum. It can be simple wound specimen in the form of pus. There can be other samples also like urine, blood, other biological fluids such as sebum, vaginal fluid, etc. So these samples are collected and then you are growing them in an artificial environment of the laboratory, which is in vitro environment. So taking the microbes which are present in vivo environment in the specimen, you are culturing them in the laboratory in vivo environment, is known as cultivation. So the bacterial cultivation basically has three purposes. Number one, to grow and isolate all bacteria which are present in an infection. Then number two, to determine which of the growing bacteria are most likely causing that infection and which ones are likely colonizers or the contaminants. And lastly, to obtain sufficient growth of clinically relevant bacteria to allow its identification and characterization. So these three steps are very important for any culture. First, you grow and isolate in the bacteria to demonstrate that there is presence of infection. Then after growth, you have to determine that which bacteria is really causing that infection and which one is either colonizer or a contaminant which has been acquired during the procedure. And then after obtaining the sufficient growth of that clinically relevant bacteria, you allow its identification and characterization. So basically, what are the ingredients which are required in culture media? Number one is water. Water is usually distilled or demineralized water which is suitable for culture media. Tap water is also useful or suitable for culture media, particularly with low mineral content. It gives a source of hydrogen and oxygen to the growing bacteria. Then you require agar. Agar is prepared from a variety of seaweeds, universally used for preparing solid media, that is semi-solid media. It is a medium which is used for solidification. And the chief component of agar is a long chain polysaccharide. It is principally used for solidifying media in concentrations of at least 2 to 3%. Therefore, 2 to 3% agar if it is present, then you are going to get a semi-solid jam-like media, jelly-like media. It melts at about 95 degrees centigrade and solidifies only when cooled to a temperature less than 42 or less. It is either manufactured as long shreds or it is commercially available in the powder form. So we have discussed water, we have discussed agar. Then there is another component which is known as peptone. Peptone is a complex mixture of partially digested proteins and is another almost universal ingredient of the commonly occurring culture media. The important constituents are peptones, proteases, amino acids, variety of inorganic salts including potassium and magnesium, and certain accessory growth factors such as nicotinic acid and riboflavin. It is a source of protein, mineral and accessory growth factors for that bacteria which we are going to culture in the culture media. Commercially, it is available as peptones or digest broth. You also have in the form of meat extracts in the form of brand name Lab Lemco. So all these can be used in the form of peptone. The other constituent is yeast extract. It contains a wide range of amino acids, growth factors and certain inorganic salts. Principally, it is used as a comprehensive source of growth factors. It may be substituted for meat extract in the culture media. So yeast extract is a substitute for meat extract to be used in culture media, providing a wide range of amino acids, growth factors and certain inorganic salts which are used as comprehensive source of growth factors for that bacteria which we are going to culture. Malt extract is another constituent. It consists mainly of maltose, which is a sugar, about 50% of maltose, starch, dextrins and of course glucose. It contains about 5% of proteins and protein breakdown products. It also has a wide range of mineral salts and growth factors which can be utilized for the good growth of bacteria which is to be cultured. Then you can also have blood or serum. They are principally used for enriching that culture media. Either human or animal blood can be used. Usually 5 to 10% of blood is used and usual concentration is 10%. Serum can also be used in certain culture media. For example, Loeffler's serum slope, which is used as a selective culture media for the growth of Corynebacterium diphtheriae. It is gram-positive bacilli which causes diphtheria in children. Now we come to classification of media. We have discussed certain features, what are the definitions, then what are the basic constituents of culture media. Now we come to classification. So first, they are based on phases of growth media. That is, it can be a liquid broth media, it can be a solid agar media and it can also be a semi-solid media. The other way to classify it is based on nutritional factors. It can be a simple media which is also known as basal media. It can be a complex media. It can also be a synthetic or a clearly defined media. Then you have special medias such as enriched media, enrichment media, selective media, indicator or differential media, transport media and sugar media. You also have certain reducing media in the form of liquid broth media, solid agar media and semi-solid media. So these are various methods by which you can classify these culture medias. First we will discuss phases of growth media. So first is liquid broth media. In broth media, nutrients are dissolved in water and bacterial growth is indicated by change in the broth appearance from clear to turbid, which is a bit cloudy. So after growth of bacteria, it becomes turbid. It is used for obtaining bacterial growth either from blood or water or for preparing bulk cultures for preparation of antigens or vaccines. The disadvantage of this liquid broth media is that growth does not exhibit special characteristic appearance in them and therefore it is of limited use in identification. And also organisms from mixtures cannot be separated with certainty by growth in a liquid media. So if solid media are used, these disadvantages can be overcome. So solid agar media is made by adding a solidification agent, which we have already discussed. It is agar to the nutrients and water. On solid media, bacteria produce discrete visible growth in the form of colonies. What are colonies? These are clones of cells originating from a single bacterial cell. So defining colony is clones of cells which are originating from a single bacterial cell. On a solid media, bacteria have distinct colony morphology and also exhibit many other characteristic features such as pigmentation, such as pigmented bacteria. If you talk about, there is bacteria, gram-positive bacteria known as Staphylococcus aureus, which produces golden yellow pigment on nutrient agar. Then there is a bacteria which is known as Pseudomonas aeruginosa. It is a gram-negative bacteria. It produces greenish yellow pigmentation on agar, and particularly this pigment diffuses in the medium. So culture media also appears to be green in color. There are other examples of culture media bacteria which produce pigment like Serratia and many more. Then after pigmentation, you can also visualize hemolysis, making the identification of that particular bacteria very easy. Okay, so for special purposes, the agar is added to the medium in concentration of 0.2 to 0.5% to solidify them. It yields semi-solid medium through which motile but non-motile bacteria may spread. Okay. Complex media is composed of indigenes such as peptones and extracts which vary in their chemical composition. Chemically defined media is composed of precise mixtures of pure chemicals such as ammonium sulfate. Then you have special media like enriched media. These enriched media are used to grow bacteria which are more exacting in their nutrition requirements. Substances such as blood, serum or egg are added to basal medium to make it an enriched medium. And mind you, this enriched medium is always a semi-solid medium. Then another type of special media is enrichment media. Enrichment media is also similar to selective media, but it is designed to increase the number of desired microbes to a detectable level. And always this enrichment media is liquid. It is in liquid phase. Then you have selective media in which there is suppression of unwanted microbes and you encourage desired microbes. An example of selective media can be LGA media, which is used for the growth of Mycobacterium tuberculosis. Then indicator media. It contains an indicator which may change color when a bacterium grows in them. The best example is McConkey agar. In McConkey agar, the colonies which are lactose fermenting, they appear pink to red in color, whereas those bacteria which are non-lactose fermenting appear pale in color. So indicator is changing the color based on the pH of that culture media. Then you have differential media. Medium that contains any ingredient that can be changed by certain bacteria in a recognizable way. Differentiation of colonies of desired microbes from the other, the colonies can be easily differentiated of two different bacteria. Sugar media, sugar fermentation reactions are carried out for the identification of most of the organisms. So there are certain sugars which are fermented by certain group of bacteria. The examples are maltose, sucrose, mannitol, glucose, etc. Transport media. Transport medium is holding medium designed to preserve the viability of organism in the specimen but not allowing its multiplication. Whenever you need to process that bacteria, the processing laboratory is far, so you require to transport that particular specimen in a transport medium so that viability of that microbe is not interfered. The viability is gained. The bacteria is live although it cannot multiply. Then there are certain reducing media for growth of obligate anaerobes. Talking about simple media, they have only the basal medium. They do not provide any special nutrient or any added ingredients for bringing out certain characteristic of bacterium growth in the culture media. The best example is in liquid form, nutrient broth, and in semi-solid form, nutrient agar. Talking about nutrient agar, it is straw-colored appearance with its composition containing nutrient broth and agar in the dilution of 2 to 3% to solidify it. It is used for routine cultures. The basal medium used to prepare blood agar, chocolate agar, deoxycholate agar, and bile salt agar. Enriched media are those media in which substances such as blood, serum, or egg are added to the basal medium for growth of certain fastidious bacteria which are exacting in their nutrient requirements can be grown on these media. And the best example of this culture media is blood agar and chocolate agar. Talking about blood agar, it is bright red in appearance and the composition of blood agar is sterile nutrient agar in addition to 5% of sterile sheep blood which is defibrinated. It is used for the growth of many medically important fastidious bacteria such as isolation of streptococci, Streptococcus pneumoniae, which is also known as pneumococcus, Haemophilus, etc. Chocolate agar is also an enriched media, but its appearance is chocolate in agar. It is not that chocolate is added to it. It is heated blood agar. That if you heat the blood agar, which contain nutrient agar and 2 to 3% of sheep's defibrinated blood, you heat it for 55 degrees centigrade for at least 2 hours. The red blood cells are lysed. Therefore, it appears chocolate brown in color. This is also used for the growth of medically important fastidious bacteria such as Neisseria gonorrhoeae, Neisseria meningitidis, Haemophilus influenzae, and Pneumococcus. Enrichment media is a mixed culture and a liquid medium. Certain substances are incorporated in that liquid medium which have a stimulating effect on that bacteria which is to be grown and inhibitory effect on the bacteria which is to be suppressed. This results in an absolute increase in the number of bacteria which we want than relatively to the other bacteria which we don't want. So it is always a liquid media. Then selective media is always a solid media. Some substances are incorporated in solid medium which have an inhibitory effect on the unwanted bacteria for which you have to suppress the growth of that bacteria. You have to add certain inhibitory effects. So this results in absolute increase in the number of wanted bacteria relatively to that of unwanted bacteria. Examples of the selective media are deoxycholate citrate agar, potassium telluride blood agar used for Corynebacterium diphtheria, Wilson and Blair medium which is used for the growth of Salmonella and Shigella. Then differential media is also a solid medium. Some substances are incorporated in solid medium which enable to bring out differing characteristic of bacteria, thus helping to distinguish between them. Such media is called as differential media. And the best example, as I told you, is McConkey agar and Tled agar of differential agar media. An important media is anaerobic media. These media are used to grow anaerobic organisms, and the examples are thioglycolate broth and Robertson's cooked meat media. The Robertson cooked meat media is a liquid media which consists of fat-free minced cooked meat in a broth with a layer of sterile Vaseline or paraffin oil over it. It permits the growth of even strict anaerobes and indicates their either saccharolytic effect or proteolytic effect by the meat being turning red or black respectively. That is, the meat is being used by bacteria either saccharolytically. If it is used saccharolytically, it will turn red in color. If it is used proteolytically, then the color will change to black in color. Sugar media helps in differentiation of bacteria. The term sugar denotes fermentable substances. So as I told you, the examples of sugars are glucose, lactose, sucrose, maltitol and maltose. They are routinely employed for fermentation tests. This culture media is a liquid culture media which contains 1% sugar in peptone water along with a suitable indicator. The advantage of using this indicator is that this indicator will change the color in specific pH. That is, if the sugar is being utilized, then the color would change from red to pale in color of the phenol red indicator. Transport media helps in transport of delicate bacteria to the laboratory for testing. So when bacteria cannot survive in the time taken for transit or may be overgrown by other commensals and colonizers which are present in the sample. So alkaline peptone water is used for transport of Vibrio cholerae in stool sample. This is an example of transport media. Then you have fungus culture media also. Sabrod's dextrode agar is commonly used media for the growth of fungus. Substances such as glucose, peptone and agar are incorporated in this solid media. It enables the growth of fungus, but the growth of fungus in the population generation time is quite long in fungus, so it takes about 1 to 3 weeks for the growth of fungus in this Sabrod's dextrose agar. So basically all these culture media which we have discussed today, they are usually available in commercial forms. They are commercially available. Like a very important example is company High Media. It is having commercially available these culture media in powder form. What you have to do, you have to just prepare it in house, and after preparation you have to sterilize that culture media in autoclave or in spacer depending on the heat requirement of that bacteria, and then you store it without disturbing in a refrigerator, and whenever you require that culture media you can utilize it for the growth of bacteria. So all these culture media are really very important for the growth of different types of bacteria, and these culture media are routinely employed for the growth in the laboratory. Thank you very much for your kind attention. If you have any doubts, you can ask. Just a word about anaerobic bacteria, how these anaerobic bacteria grow. These anaerobic conditions or anaerobiosis of within the laboratory can be attained. For the anaerobiosis, it can be attained either by production of vacuum, by displacement of oxygen, by displacement and combustion of oxygen, by absorption of oxygen by chemical or biological method, or using certain reducing agent. As we all know that these anaerobic bacteria will grow only in absence of oxygen. That is anaerobic condition, and these are the methods by which you can grow these bacteria in different methods or forms. So for the production of vacuum, cultivation in vacuum was attempted by incubating cultures in a vacuum desiccator, but unfortunately it was proved to be unsatisfactory. Therefore this method is not used now, but theoretically you need to know that by production of vacuum you can grow these bacteria by anaerobic method. Then displacement of oxygen. Displacement of oxygen by inert gases such as hydrogen, nitrogen, carbon dioxide or helium can sometimes be employed. By this method, oxygen can never be completely removed. The best example is using a candle jar is a popular method, but it is an ineffective method. What has happened? A lighted candle is kept in a large airtight container which is loaded with different inoculation plates. It is expected that the burning candle will use up all the oxygen before it is extinguished. Although in this method some amount of oxygen is always left behind. So the best example of this is candle jar, displacement of oxygen by candle jar. Then another method for producing anaerobiosis is by displacement and combustion of oxygen. So this anaerobiosis is obtained by Macintosh Fielday's jar, and this method is the most reliable and widely used method. How? This jar consists of a stout metal or glass jar with a metal lid which can be clamped airtight with a screw. The lid is fitted with two tubes with tabs, one acting as an inlet for the introduction of gas and the other as outlet. The lid also contains two terminals that can be connected to an electrical supply. A catalyst, that is alumina pellets coated with palladium are used, is suspended under the lid by stout wires connected with the terminals to heat the catalyst for its activity. Nowadays, catalyst without heating at room temperature is also available, which can be utilized in this Macintosh Fielday's jar. The culture plates are inoculated with the specimen and they are placed inside the anaerobic jar with an indicator and the lid is clamped tight. The outlet tube is connected to a vacuum pump while inlet tube is closed. The air inside is evacuated through vacuum and the outlet tube is then closed. Hydrogen gas is passed through the inlet tube till the reduced atmospheric pressure is brought to the normal atmospheric pressure, that is 760 mm of mercury, which is monitored on the vacuum gauge as 0 point. Then electrical terminals are switched on for heating of the catalyst. It helps to combine hydrogen with the residual oxygen to form water. And the indicator which used is reduced methylene blue in the jar. It remains colorless in anaerobic condition but will turn to blue or violet in color on immediate exposure of oxygen. So this is the most reliable and best method by displacement and combustion of oxygen. Then you have certain chemicals for the absorption of oxygen like pyrogalol. Alkaline pyrogalol absorbs oxygen. It was introduced as early as 1888 by Buchner. There are large tubes which are containing solution of sodium hydroxide and pyrogallic acid placed inside the airtight jars, which may produce the anaerobic condition. The other is chromium and sulfuric acid. A mixture of chromium and sulfuric acid is used for producing anaerobiosis. These two chemicals react in presence of oxygen and produce chromous sulfate. So oxygen is being absorbed. Then there is absorption of oxygen by certain chemical methods, like these days we are using gas pack. This is the most reliable method which is being used these days. It is available commercially as disposable packets containing pellets of sodium borohydride, cobalt chloride, citric acid and sodium bicarbonate. It is a simple technique which is widely used for preparing anaerobic jars. These chemicals generate hydrogen and carbon dioxide when the water is added. The hydrogen combines with the oxygen in the presence of a catalyst. So gas pack with water is kept inside the airtight jar which is containing the inoculated plates, and the lid is closed tight. So there is complete absorption of oxygen in the jar and total anaerobiosis is maintained. Then absorption of oxygen by certain biological method. Although this method is also not very reliable, but for theoretical point of view you need to know. It has been attempted by incubating aerobic organisms along with anaerobic bacteria. That is, you can take two blood culture plates. One is inoculated with aerobic bacteria such as Pseudomonas aeruginosa and other with the specimen of anaerobic bacteria. And these two plates are inverted and placed over one another, and they are sealed along the rims and they are incubated. It is always expected that the aerobic bacteria will utilize all the oxygen which is present within the culture plate and in turn will release carbon dioxide due to its metabolism, which will create an anaerobic environment. But this method is very slow and ineffective. Again, it is important for theoretical point of view. Then certain reducing agents are being used. Oxygen in culture media can be reduced by certain agents like glucose, thioglycolate, cooked meat pieces, cysteine and ascorbic acid. Based on this principle, two widely most employed anaerobic liquid culture media are thioglycolate broth and Robertson cooked meat broth. Robertson cooked meat broth contains Newton broth and pieces of fat-free minced cooked meat of ox heart. Principle of this is that unsaturated fatty acids which are present in meat utilize the oxygen for auto-oxidation, and this reaction is catalyzed by hematine which is present in meat. Then glutathione and cysteine, both are reducing agents. They are also present in meat, also utilize the oxygen. Sulfhydryl compounds which are present in cysteine also contribute for a reduced oxidation reduction potential. How it is done? Before inoculation, the medium is boiled in water bath for 80 degrees centigrade for about 30 minutes to make it oxygen free. For strict anaerobiosis, the surface of cooked meat broth medium may be covered with a layer of sterile liquid paraffin, and the specimen is inoculated inside it. It is incubated. How interpretation would be done? Saccharolytic anaerobes, like for example Clostridium perfringens. It is an anaerobe which is saccharolytic. It will turn the color of meat pieces into red, while it becomes black in case of proteolytic anaerobe. The best example is Clostridium tetani. Then you also have a anaerobic chamber. It is an anaerobic incubation system. It provides oxygen-free environment for inoculating culture media and for their incubation. It is fitted with airtight rubber gloves to insert hands for working with specimen. These anaerobic chambers contain a catalyst, a desiccant, hydrogen gas, carbon dioxide, nitrogen gas and an indicator. It is simple like a biological safety cabinet which is airtight, containing all these catalysts, desiccants, hydrogen gas, carbon dioxide, nitrogen gas and an indicator. And there are certain holes made fitted with rubber gloves through which you can put your hand inside and you can have a working environment inside. This will produce absolute anaerobiosis. Okay, so these are the basic methods, anaerobic methods which are used for producing anaerobiosis. So today friends, we have discussed different type of culture medias, how they are prepared, what are the constituents, how it is sterilized and for what all they are being used. Thank you very much for your kind attention. If you have any doubts, you can ask me.